note, I really like to acknowledge the project team that beside me was Gunnar Günther, the project leader, and Christoph Lange as the principal investigator, but also the large number of physicians and scientists in the different health facilities. It's not a project that is done by just 2 or 3 people. And lastly, on the last line, we see the names of Cordula Ehlers, Bram Prins, and Nelleke Smitsman, who really were instrumental in support in the administrative area and data management. So, from resistance to treatment regimens, and that was quite a work in our clinical cohort, in our data analysis. So first of all, we wanted to see if patients started with a standardized regimen or an individualized regimen. And for that, we defined a standardized regimen as one with a fluoroquinolone, a second-line injectable, proteonomide or ethionomide, cycloxanone or PAS, and pyrazinamide. We could have chosen for slightly different definitions, but the findings would be the same. Because actually what we see that in patients from low incidence countries, the large majority starts with an individualized treatment already, not a standardized regimen. While in high incidence countries, the large majority of patients has to start with a standardized regimen. And that is important for the treatment outcomes, which I will discuss later on. So then with what type of regimens did the patient start? This is a very busy slide, but I will take you through it. The first thing to acknowledge is that on the y-axis and the x-axis there are the individual drugs, and they are exactly the same on the 2 axes. So ethambutol here and ethambutol here. So those axes are the same. As soon as there is a dot, that means that those 2 drugs are used in combination. So for example, PAS here is used in combination with proteonomide. If the dot is red, then there is a strong correlation. So if one drug is used, the other drug is also used more likely. And if the dot is blue, then there is a negative correlation indicating if one drug is used, then the other drug being used is less likely. And the bolder the dot, the stronger the correlation. So that is the background of these graphs. But without going into detail, what we can see in the low incidence country is that every drug combination has a dot. So every drug is used by somebody in a combination with any other drug. So there's a lot of ways how regimens can be constructed. When we go to intermediate and high incidence countries, we all of a sudden see wide gaps in these graphs. So that means that for these drugs, there is nobody who uses that drug in combination with another drug. So the possibilities of constructing an appropriate treatment regimen is much more limited in high incidence countries, and to a certain extent also for the intermediate incidence countries. We were especially interested in the use of pyrazinamide in the starting regimen because that remains a debate. So we first look at how many patients actually used pyrazinamide in the starting regimen, and that is denoted by the black bars. And then we assess of these patients how many patients actually had drug sensitivity tests for pyrazinamide. That is the light green bar. And of those tests, how many showed sensitivity towards pyrazinamide. And if you put these 2 together, we can calculate how many patients that use pyrazinamide in their starting regimens actually do so with a proven susceptibility. So in low incidence country, we see that 55% of the patients are using pyrazinamide in the starting regimen, but that only 65% of them have a proven susceptibility. But what happens if we look in high incidence countries? The large majority of the patients used pyrazinamide in their starting regimen, and that is definitely a reflection of this standardized regimen. But hardly any of the patients using pyrazinamide do so with a proven susceptibility. So more or less, pyrazinamide is given blind. And that really questions the use of pyrazinamide in a standardized regimen. What we try to do next is actually capture the treatment changes, because we talk a lot about starting regimens, but therapy is long and people changed therapy. So we try to capture each and every treatment change, and we ask the physicians to record every start and stop date of a drug, which resulted in over 12,000 different treatment records for 377 patients, because 3 patients of our cohort never reported any treatments. So few patients, huge amount of treatment records, making analysis of them a rather big task. But if we look when the treatment changes happen, they happen both in the intensive phase and in the continuation phase. And this circle here identifies only 11% of the patients do not change their treatment throughout the full treatment course. Throughout the treatment course. So 90% of the patients change their treatment throughout the treatment course, and this is corrected, of course, for the mandatory change in second-line injectables, which questions the approach of analyzing treatment efficacy according to the starting regimen. So with regards to treatment, we see marked difference in the use of standardized regimens by TB incidence, and the use of pyrazinamide in the starting regimen is often not guided by drug sensitivity testing. And as just said, very frequent treatment changes make analysis of treatment efficacy complicated.